So what we'll be discussing at this time is polyspecific AHG. So we understand the IAT and we understand the anti-human globulin testing methodology. We understand the history, where it was coming from. Now we understand in parts how it came about. So we're going to now try to dissect when these animals are infused. How do we get polyspecific? How do we get monospecific antibodies? So that is what we're going to be getting into at this time. So when we talk about polyspecific AHG reagent, these contain antibodies to human IgG and to C3d complement of the human complement. All right. Other anti-complement antibodies such as anti-C3b may also be present. Therefore, it is useful to note this information. So we have IgG and we have C3b, or in some instances C3d, or both. But the two most common are the IgG and the C3d. When we talk about poly, I want you to think about multiple. So think about polygamy, which means more than one. All right. And we're not promoting polygamy, we're just using the word poly so that we can have an understanding of the amount of counts, right? So that's more than one. So let us just look at the polyspecificity, or the polyspecific AHG. So these are coming from rabbits, polyclonal. You may hear that term very frequently. And these contains IgG and anti-C3d. And it may contain anti-complements and other anti-immunoglobulin antibodies. Yes, you hear us saying anti. It is not your auntie that lives in the country. We are talking about anti, things that are against, right? So sometimes you have to use what is against in order to find what is with. All right, so that is just the idea that I want to bring to your attention. Then we have rabbit, or murine monoclonal blend, which contains a blend of rabbit polyclonal anti-human IgG and anti-C3d, is a murine monoclonal antibody for the IgM. All right, so we have different categories, but we have poly and we also have what we call monospecific. So the monospecific AHG reagent contains only one antibody specificity, either the IgG or the antibody to a specific component of complement. It can be C3b or it can be C3d, or whatever component that is they want it to be specific to. But the licensed monoclonal specific AHG reagent in common use are those that comprises of anti-IgG and C3b or C3d. So those are the ones that we find most frequently in the laboratory and in the transfusion medicine and in the whole scope of laboratory practice for routine activity. However, when you're doing more specialized testing, then that is where the specific antibodies can come in handy, especially when you're doing antibody identification, especially when you're performing elution techniques, especially when you're looking for something that is not common occurrence. Now, the anti-IgG reagent labeled anti-IgG contain no anti-complement activity. All right, so the anti-IgG does not have any complement. anti-IgG reagent contains antibodies specific for the Fc fragment of the gamma heavy chain of the IgG molecule. So the anti-IgG has no complement in there. It is specific for the Fc fragment of the gamma heavy chain of the IgG molecule. If not labeled gamma heavy chain specific, the anti-IgG may contain anti-light chain specificity and may therefore react with cell sensitized with IgM. It can also react with IgA, and it can also react with IgG. So if it has anti-light chain specificity, it can react with IgM, IgA, and IgG. Now, when we look at the anti-complement reagent, such as the C3b and the C3d reagents, they are reactive against only the designated complement component and may contain no activity against human immunoglobulins. All right, very good. So you're getting the big picture. So the monospecificity anti-complement reagents are often a blend of monoclonal anti-C3b and monoclonal anti-C3d. All right. So now you may be wondering, how do we go about preparing the anti-human globulins? So the classical method of AHG production involved the injection of the human serum or purified globulin into a laboratory animal, such as rabbits, which have been utilized in the past and still in today's dispensation. The human globulin behave as foreign antigens. The rabbit immune system is then triggered and an antibody to the human globulin is produced. For example, human injected IgG into the rabbit result in anti-IgG production. Simple as that. Just think about it like immunization. You get something that you were not previously exposed to, the body develop antibodies against it. It's the same principle taking place in a foreign animal, right? So we call those a lab animal. So this is a response that naturally occurs. When we have polyclonal antibodies, they are a mixture of antibodies from different plasma cell clones. So you know, you have plasma, you have different cell lineage, and you have the different fragments and fractions. So what is happening in this situation is that you have one lineage, another lineage, and you combine those two lineage or more, and that there will generate a poly activity. So when we think about monospecific, we want to think about monogamy. When we think about polyspecificity, we want to think about polygamy. And we're not promoting either or. We're just trying to get your minds wrapped up so as you understand exactly what is taking place. So the polyclonal antibodies are a mixture of the antibodies from different plasma clones. Good. And the resulting polyclonal antibodies recognize different antigenic determinants, which we call epitopes. All right, or the same portion of the antigen but with a different affinity. So we're going to look at the word affinity and avidity in order to understand and get a better appreciation of what is happening. But before we get there, we have hybridome technology that can be used to produce the monoclonal antiglobulin serum, and the monoclonal antibodies are derived from one clone of plasma cells and recognize a single epitope. Now, polyclonal specific AHG is important to us, because sometimes you are in a situation where you just want to know if something is positive or negative for either or both, and a poly reagent comes in handy. But when you're trying to differentiate, that there is where the mono becomes important. So polyspecific AHG can be made using polyclonal or monoclonal antibodies. So you can take the monoclonal, combine together, get polyclonal, or you can just create your own polyclonal from start. The two types of antibodies production processes are very different from one another, yielding two very different advantages and also disadvantages. So the polyclonal AHG production, the polyclonal AHG is usually prepared in rabbits, in contrast with the early production method, in which a crude globulin fraction of serum was used as the immunogen. Modern production commences with the purification of the immunoglobulin from a large pool of normal sera. Conventional polyspecific antiglobulin reagents are produced by immunizing one colony of rabbits with immunoglobulin IgG antigen and another with the C3 antigen. Because of the heterogeneity of IgG molecules, using serum from many donors to prepare the pool IgG antigen to immunize the rabbit, and the pooling of the anti-IgG antigen to the pooling of the anti-IgG from many immunized rabbits is essential in producing the polyclonal reagent for routine use that are capable of detecting the many different antibodies that exist. And this is just the tip of the iceberg as to what the AHG polyclonal production looks like and what the AHG monoclonal production looks like. It gets deeper, but at this time, let us take a break and then get right back into the class session. Let us take a pause.